Aptamers are short oligonucleotides that are capable of acting as drug carriers while possessing immense therapeutic properties. These molecules have high target specificity and affinity, enabling drugs to be released in a specific target region with minimal toxic ramifications.
Aptamers are often referred to as synthetic antibodies because of their ability to recognize a high degree of molecular targets. They are more versatile relative to antibodies due to their immunogenicity and their ability to remain unaffected by physiological disruptions. Once they're in close proximity to the target molecule, they structurally conform to bind strongly to their target.
We can precisely control the biological function of aptamers because of their ability to be synthesized chemically. Being able to control its synthesis results in properties such as increased purity and a beneficial pharmacokinetic profile.
The most common type of aptamers are single-stranded oligonucleotides which have been selected and optimized for its specific function. This is done by using a screening process called systemic evolution of ligands by exponential enrichment, abbreviated as SELEX. SELEX uses a cycling system to select aptamers that bind most preferentially to a purified protein target of choice.
As the aptamers are amplified through PCR, the affinity they possess for their target is increased, resulting in millions of copies of a single aptamer with the strongest binding potential being produced. Coupled with the recent development of cell-based selection techniques, aptamers can be used to target cell surface proteins in their native state. These aptamers can be further manipulated to bind to a drug of choice, which can act as a cargo.
So now that we've given a little background to aptamers and their ability to target specific proteins on the cell surface, let's talk about the biophysics behind the role of aptamers in drug delivery.
Cell-based disorders or diseases require drugs to be physically delivered to the disease sites, such as the regions of intracellular or extracellular proteins, mitochondria, lipids, or even membrane proteins.
General drug delivery methods rely on passive diffusion across the membrane barrier, which can be difficult. Brownian motion is the driving force for this dissipative movement on nanoparticles. This type of diffusion occurs by establishing a concentration gradient from the extracellular region to the intracellular region. However, this is not always possible due to molecular constraints such as imposed charge or steric bulkiness.
Membranes consist of phospholipids that have a hydrophilic head group and hydrophobic tails. These phospholipids spontaneously assemble to form a bilayer, which is driven by the hydrophobic effect. The hydrophilic head groups interact with the solvent, and the hydrophobic tails cluster to form the core, maximizing solvent entropy by minimizing the formation of clathrates.
Since aptamers contain a negatively charged phosphate backbone, they cannot passively diffuse across the hydrophobic core. Thus, binding to an extracellular cell surface protein is required for cell entry and adequate drug delivery by releasing the cargo contents of the aptamer. All cell surface proteins cycle intracellularly, which means that anything bound to the embedded protein will be internalized into the cell.
Once aptamers recognize their protein target, they will bind through complementary base pairing to produce secondary structures with the protein. Further folding of these secondary structures results in the generation of tertiary structures. This increases the variety of electrostatic interactions that aptamers can participate in with their target, including van der Waals forces and hydrogen bonding, which offsets the loss of entropy. Formation of this tertiary structure leads to a stable aptamer-target complex.
Fick's law is relevant in this case because the aptamer has to diffuse freely in order to reach the target site, where it binds to the protein and is internalized by the membrane. As shown on the screen, J is equal to negative D change in C over change in X, where J is the diffusion flux, which refers to the amount of substance diffusing per unit area per unit time, and D is the diffusion constant, which is equal to a unit length squared over 2 multiplied by time.
Thus, diffusion rate equals surface area times concentration gradient divided by diffusion distance. Because the plasma membrane surface area and diffusion distance can be manipulated during in vitro experiments but cannot be modified in the human body, in order to increase the diffusion rate, the concentration gradient should be maximized and aptamer molecules should be designed to be smaller sized.
Shown in this diagram, we see a single-stranded RNA aptamer and a cell surface protein in its native state. When placed in close proximity to the native protein, it will undergo electrostatic interactions like hydrogen bonding and bind non-covalently to the native protein. There is a loss of conformational states, also referred to as a decrease in omega, when binding. However, this decrease in the degrees of freedom is offset by a decrease in enthalpy due to preferable electrostatic interactions. As denoted in the equation S is equal to Kb ln omega, the entropy of the protein aptamer complex will be lower than that of the aptamer itself.
The aptamer binding to the protein can be denoted by the equilibrium expression free energy equals negative RT ln Kd, where Kd is the dissociation constant. A system will change its state, in other words its conformation, if it lowers the free energy. Therefore here, we want to minimize the Kd to maximize the free energy difference. Although there is a decrease in entropy due to the loss of conformational states of the aptamer, this is compensated by the decrease in enthalpy, meaning that more electrostatic interactions are present. It can also be mentioned that once the aptamer enters a cell, dissociation will occur and the cargo will be released to deliver the drug effect. This results in a lower free energy state due to the entropy gained in the system.
Although the conclusions that we've made in this presentation are based on in vitro data, we believe that the translation of this approach to human patients in vivo is a realistic goal for the near future.